Well, good morning, everyone. Thank you for joining us today. I'm John Tillotson. I'm serving as the host of our session today. This is the first episode of a planned series of educational sessions where we will explore examples of solving water mysteries with modern DNA science and technology. So thank you so much for joining us today. I'm really excited to be facilitating this discussion. I think you're going to be quite pleased and inspired by the story today. Let's begin by introducing our speakers. First is Linda Rost, who is quite an amazing science teacher, in my opinion, at Baker High School in Baker, Montana. Linda has quite a track record of inspiring students to do great things in science. And it's been a real pleasure working with Linda over the past year or so, working on water quality issues at Baker Lake, which is a nearby freshwater recreational lake near the high school. One of Linda's notable accomplishments is winning Teacher of the Year in the state of Montana, which is pretty cool. I don't know how you do that, Linda, but apparently you do. And also with us is Hunter Bailey. Hunter is in the graduating class this year at Baker High School. Congratulations, Hunter, on graduating, which I understand occurred just this past Sunday. And Hunter was in Linda's advanced placement biology class this spring, which is where he got involved in the water quality issues at Baker Lake. And his findings are quite revealing. So Linda, let's begin with you. Can you tell us a little bit about what you do as a science teacher at Baker High School? And how did you get involved bringing water quality issues at Baker Lake into the classroom?
Yeah, so I teach mostly the biology and chemistry classes at Baker High School, and it's a small school. We're about 100 miles from the closest Walmart and McDonald's. And I've been teaching here for 10 years. So I teach biology, chemistry, AP biology, anatomy and physiology, and then I teach a science research class. And the science research class is where Hunter did most of his work on this project. And I've been teaching that since I started teaching, and it's really shaped who I am as a science teacher. In that class, students are able to design novel experiments on anything that they're interested in, and then they work all year working on their study, and then they compete at science fairs and research competitions. So that's really allowed me to kind of like stretch outside of the normal science teacher realm and be able to address topics that are interesting to students and that matter to the community. So the way that this started was 2 years ago. I had a sophomore come in and ask questions about the lake. People had been getting sick after swimming in the lake, and a lot of the kids were talking about it and wondering about it. And so the student wanted to sample the lake and see what was going on. And so I let him. And I had been, if I back up a little bit, I had been doing STEM camps for kids in like K-8 over the summer for the last several years. We had been sampling bacteria in the lake then also, but just kind of at a small scale with little kids. So this student wanted to sample it and see what was going on. He took some samples. The plates were just covered in bacteria. It was like a lawn of bacteria from 1 ml of lake water, which if you've grown bacteria, that's a ton of bacteria. So that's when I started kind of wondering, like maybe something is going on. So then I had all of my sophomores take samples from different spots all over the lake, and we grew them. And the same, I thought maybe it was a fluke with this initial student, but the same thing happened. All the plates were full of bacteria. So that's when I started to suspect like something might be wrong compared to when we would do this with the STEM camp years before. There was a difference. So that kind of started the first research project. I had 2 students last year work on testing the lake and kind of determining the nature of the bacteria. And we did some genetic testing of the bacteria for that project. And then Hunter this year joined, and he worked on trying to determine the source of the fecal contamination. So we did identify that it was E. coli and that it was a high level of E. coli. Sometimes it goes above the DEQ standards. And then the coliforms are also very high. So Hunter will show you that in his stuff. So he was looking at what could be causing this, and there could be many causes, but he focused on looking at what was going on the golf course. So that's kind of the evolution of the project, and it's just kind of flowed as we find new information. And we also have been able to partner with the county. So they actually funded the supplies and the testing for this project, which has been incredibly helpful because with a high school teacher budget, there is no budget for any of this. So it's been amazing to be able to partner with the county and the community and get some help that way. We also partnered with DEQ, and they helped us with supplies and information and all of that.
Yeah. Yeah. No, thank you for that. It's really fantastic to see how you can incorporate teaching your students in solving real world water quality problems is really a great model, I think, for us to take a look at and for others to try to follow. And Hunter, how about you? How did you tell me a little bit of backstory on how you got involved in this? I know this was part of your class, but you were the only student that it seems really got into this. Is that correct?
Yeah, that is correct. Kind of started, I guess. I've always loved science. It was always my strongest suit. And so I kind of just took the jump at it when the opportunity arrived. I know that nobody was going to do it, so I kind of took a jump at it, kind of knowing a little bit about it, but kept learning with the help of my science teacher. And then, yeah.
And apparently you did pretty well. Can you tell us a little bit about the Montana State Science Fair and what that process was like?
Yeah, so at Montana State Science Fair, right in the morning, go set up, but you kind of get judged. Can't remember the time, but you get judged for like a couple hours. You have 3 or 4 that judges come up to you. And then to advance to the finals, you have to get a gold ribbon. I got the gold ribbon. And then when you get to the finals, kind of everybody that didn't make it leaves. And then more judges come up. There's not really a set how many judges. So I had like 4 others come up to me wanting to know the project. And after that, then you just wait to see your results the following day. One state and one best overall STEM and the ISF finalist.
It's really impressive. Must have been pretty exciting to win that. So you won the grand prize at the State Science Fair in Montana with this project.
Yes.
Well, congratulations on that. It's a great way to finish your high school years.
Mm-hmm.
So
Yeah.
with that being said, why don't we go ahead and Hunter has some slides prepared for us to take us through his research project.
All right. My project's on microbial source tracking of fecal contamination in the Baker Lake from recycled wastewater use for the Lakeview Golf Course irrigation. Some people like to thank the Fallon County and the commissioners, Montana DEQ, microbe detectives, Energy Labs, Fallon County Sanitarian, the Lake Advisory Board, and my science teacher, Linda Rost. This project came to life again in the summer of 2022 when the reports of swimmers getting sick after recreational use of the Baker Lake in Baker, Montana. These illnesses were reported to the Fallon County Health Department. These illnesses include swimmer's itch and gastrointestinal illnesses. DEQ identified a toxic algae bloom in the summer of 2022, but the county tests were negative. In 2022, a student group partnered with the county and DEQ and found that the lake had way over the recommended level of coliforms and high levels of E. coli, which could be causing the human illness. But the sources of contamination were yet unknown. But one possibility could be the runoff from the irrigation of the nearby golf course. And this brings us to the nearby golf course, which is the Lakeview Golf Course. The upper and lower lakes are 8 meters lower in altitude compared to the golf course. The city of Baker chlorinates the lagoons with 0.5 pounds of chlorinated gas per 100,000 gallons, but surface water should receive a dosage of 5 to 10 milligrams per liter. The golf course must follow irrigation rules for the recycled wastewater. And as you can see on to the far right, the little map, you can see that the golf course is over here, the irrigation pond is here, the lagoons there, and then the upper and lower lakes right here. This brings us to the recycle wastewater irrigation rules and classes. A class A is what the golf course has to be. It has unrestricted golf courses, parks, schoolyards, and other testing is required. You can have an average of 2.2 CFU per 100 mil, and then the max you can have is 23 CFU per 100 mil. The county does state that it is a Class C, but again, Montana DEQ says it's a Class A. Class C states that it has to be used for landscape irrigation. You can have an average of 23 CFU per 100 mil, and then a max of 240 CFU per mil. The last sample taken from the Lakeview Golf Course effluent was on October 2nd, 2023, and that was 1986.3 CFU per 100 mil. The levels surpass Class A, B, and requirements, and Class C requirements for call forms. But this brings us to our wastewater compliance. An operation plan for disposal of wastewater effluent to be used for Lakeview Golf Course irrigation was received by the Montana DEQ and denied. Coliform tests show the total coliform count and the effluent to be over 2,400 CFU per mil. The effluent does not comply with the maximum coliform concentration for even Class C, but again, the golf course is open to the public. And this brings us to the runoff from recycled wastewater. Reports of high coliform counts in the irrigation water. That is possible that the recycled irrigation wastewater from the Lakeview Golf Course is running off into the lake. On September 4, 2023, we had a severe storm that dumped more than 2 inches of rain, creating severe flooding across Fallon County. When there's video evidence of this. And again, there's storm drains for the rainwater, but it plugged. Storm drains only have a max capacity, how much they hold, and they got plugged after a while, so you could see the water running off into the lake. But this event does give evidence that storm water runoff from the golf course does run off into the lake. Determining the source of contamination, I use a tool called microbial source tracking, and that is a tool that can be used to obtain whether the fecal contamination water came from human, animal, or even both. I use 2 methods. The first one is fecal indicator bacteria test, or FIB, and that is used for stormwater and freshwater samples for coliform E. coli and fecal indicators. The second one is the gene markers, and that is in stormwater and freshwater samples, and that can indicate whether the gene is associated with human or animal feces. And the human fecal gene we're looking for is the HF183. There's a third one. I didn't use it, but that is biotracer, and that is when you use a specific E. coli strain, you put it on the surface of the soil and test other locations to see and detect movement. Again, FIB tests and gene marker MST methods were used in this one. And this brings us to our part 1, the fecal indicator. Partnered with DEQ to conduct fecal bacteria tests using Colfer and Quantitre 2000 using DEQ protocols. The samples were taken from all sites from 8/31/23 to 9/11/23, and that's A, B, D, and E, and that'll be the map right here. In the middle, as you can see, A is over here, B is right by the Iron Horse Park, B is by the other beach, and then E is the upper lake. And then after flash flooding on the rainstorm on 9/4/23, we tested it on 9/5 and 9/11/23 And those were the puddles between the golf course from the southern end of the Baker Lake, and that's the T1, T2, and T3, and that is the map on your far left side. And then if we jump over to the little samples from our trays that we took, these are the Calher and Conte tray 2000s. The yellow confirm the presence of coliforms, but when you turn off the lights and take a UV light to them, and they glow with fluorescent, it confirms the presence of E. coli in our samples. And this brings us to our results of part 1. If you look over here onto this graph, you can see that all the coliforms jumped and were pretty much maxed out for our lake here. The yellow line is a statistical threshold. You can have one that jumps over, but you can't have another one that jumps over that line. And then the red is our geometric mean limit. You can't have any that jump past this line, else you kind of have to shut down the whole lake. And as you can see, that Gazebo Beach and the upper lake are pretty close to jumping this line. Again, you only need a little bit more, and then they have to shut down the lake. And then this brings us to our runoff testing in the of puddles. As you can see, our T2 through and T3 and T1. As you can see on T2 on 9/5/23 after the severe storm on 9/4/23, you can see that the E. coli was maxed out in that test. And in T1, you can see there's a high range of E. coli. Same with T3. We didn't have a T1 on 9/11/23 due to evaporation of that puddle, but T3 went up in E. coli. And it's kind of weird because you shouldn't have E. coli this high in just the middle of the field again. This is just our daily tests, average or max. As you can see on site A and D, they were maxed out 9/5/23, and this was again after 9/4/23 down here. None of the other tests really jumped as high as the site A and D again. This brings us to our part 2, our ColScan Easy Gel. A subsample of 1 ml was pipetted into a ColScan bottle, treated on a ColScan Petri dish, and incubated at 37 degrees Celsius for 2 days. If you look to our right, this is a kit that we use. And as you can see, the blue, dark blue, and purple are E. coli, and then pink or red are other coliforms. And then this brings us to our results in part 2. As you can see, these are our Easy Gel plates from our different sites. As you can see on 8/31/23, we have 53 colonies. Each one is a colony on the plate, and you have to hand count it by hand. Not many coliforms on that one. And then again on 9/5/23, we had a 34 number of E. coli colonies on that plate. Not many plates had like what site D had, but they still showed E. coli and had a large number of presence of coliforms. We jump to our right here, as you can see on site D, 8/31/23. You can see each little individual E. coli colonies on that plate. And this brings us to our part 3, our metagenomics testing. Metagenomics testing was done on 2 1-liter samples from the north and south end of Baker Lake by microbe detectives, along with the HF183 microbial source tracking. We found in the north sample that there were 91 bacterial strains identified with 2 genes for antimicrobial resistance, along with 12 genes for violence factors. The 2 genes for antibiotic resistance are tetracycline and fosfomycin. And then you had your violence factors that included the bacteroids, Virgilus, and that is, these bacteroids indicate that there is fecal bacteria in the lake. But the AHF 183 test was negative for both samples. We just didn't, they could be out there, but we didn't pick it up. I mean, the lake is very vast and broad, so we just didn't pick it up in the samples. And then in the south end, there were 45 bacterial strains identified with 2 genes for antimicrobial resistance, along with 3 genes for violence factors. The 2 genes for antibiotic resistant are aminoglycoside and macrolide. And again, you had bacteroids in this this sample as well, which indicate bacteria. And then this brings us to our part 4, our golf course effluent. The results from summer testing of the Lakeview Golf Course effluent. And again, this was obtained from the Fountain County Sanitarian. Coliform levels were well above the Class A requirements and even above Class C requirements for most of the time. If we take a look to our right on the graph here, it shows the Class C, which is the yellow line, and Class A and B, which is the red down at the bottom. As you can see, that almost every test surpassed every class. The only test that didn't surpass Class C was on 6/26/23. That didn't surpass Class C, but surpassed the Class A and B, which is the golf course. But the only test that had a presence of E. coli was on 8/08/23. And this brings us to the conclusion. The one source of fecal coliforms in the Baker Lake is the golf course irrigation of wastewater. There could be other sources of contamination, but this is the main one. I recommend the city treating the lagoons with more chlorinated gas at the recommended level of 5 to 10 milligrams per liter, and continuing to test the effluent weekly for coliforms. I also want the county to use the biotracer that I couldn't use on this test, and they could put it there and watch to see if the E. coli does run off into there. Just watch it. And then I also recommend the Fountain County Commissioners to keep supporting and funding this future of this project for this ongoing issue, and then to try to eventually resolve this issue. And that's it. Any questions?
Yes, so for the audience listening in, if you have a question, please go ahead and in the Q&A, you can post your questions if you have any. While we're waiting on that, I'd like to kick it off with one question, which is, what kind of a response are you finding with the local community regarding your research here?
I mean, the commissioners, all but one took it pretty good, I think, but they know that's an issue and they want to resolve this issue. But I know the community, well, most of them are kind of worried about it because we do have people, it's heavily used during the summer. I mean, you have tons of boats and our beaches are filled with kids. But I know that the public is aware about this. I mean, it's been posted in the newspaper, and hopefully they're concerned about it, but I'm pretty concerned about it just because you have these antibiotic resistant genes that, you may not catch, but if you do, again, the gastrointestinal illnesses, when we look back on the antibiotic resistant genes, these ones are to treat gastrointestinal illness, and they treat acne and tonsillitis, and this can relate back to what they were finding in that summer of 2022.
Right. Linda, anything to add? I'm real curious about the reaction in the local community, and it's the public, the government officials. I'm thinking of also the golf course itself is a little bit under the microscope in this project, so to speak.
We had a meeting with the commission and Hunter presented to them, and so they have all the information, and so the next steps are just to chlorinate the lagoon wastewater or find some other solution, and then continue testing the effluent to make sure that it's low enough, because it's about 100 to 200 times too high. And we also talked about a lot of other interesting things, like they asked about whether the bacteria could accumulate in the soil, since the lagoon has been used to irrigate the golf course for like at least 20 years. So who knows how long this has been occurring? Is it building up? Is that what caused it to affect the lake so much? It could also be the drought. We've had a drought the last couple of years. So those are some questions they had. So I think we're kind of really at this point where we're starting to answer some of the questions, and then we're generating more questions, and that's been really fun. So the community is definitely excited and involved and curious about it.
That's really great to hear. So we have one question coming in. I'm going to read it, and I might go ahead and answer this one. How do you think the increased levels of coliform and E. coli affect the blue-green algae and harmful algae blooms that showed up in the lake? So do either of you want to take a crack at that, or would you like me to do that?
Go for it.
Okay. And well, Hunter?
Yeah, you can go for it.
Okay. Yeah. So, yes, it does absolutely relate to the algae blooms. And the reason is it has to do with excess nutrients getting into the water stream, specifically nitrogen and phosphorus compounds. Typically in freshwater lakes, it's a phosphorus concern, and in marine environments, coastal areas, it tends to be more of a nitrogen concern. But in both cases, when you have excess nutrients getting into the lake, it creates an overgrowth situation, and that's how the green algae, which is most likely cyanobacteria, which forms, it's one of the leading water quality challenges across the developed world. It's particularly a problem in agricultural areas. So you can get that from fecal. Whenever you have animal or human fecal contamination, you are also overloading the system with these nutrients. Other sources, however, it's not the only way to get the toxic algae blooms. Other sources would be fertilizer, which could be applied on the golf course, or it could be from agricultural applications of fertilizer. So the combination of the fertilizer runoff into the lake and other fecal sources, whether it's from the golf course itself in the recycled use of the wastewater effluent, or also if there's any agricultural runoff that may exist where animal waste is running off into the lake, can be a contributing factor to these algae blooms as well. But that's a really good question, and it's directly applicable to what we're talking about. Okay, another question. How do you think the increased levels of coliform and E. coli affect the blue? Oh, okay, that's the same question. We just answered that one. Any other questions from the audience today?
I see another one on there from Tiffany Wyden. She's from the DEQ. She's been helping us.
Okay, let's see. I'm sorry. Okay, did you plan some of your sampling events when you knew that precipitation events might be occurring so you could track runoff?
Do you want to answer that one, Hunter, with the flash flood?
Yeah. Again, last year, we went out right after the severe flooding and tested it, and then we also did the spring after it downpoured it for a few days. We went out there and took samples, and I don't know, what was it, last week? I was signing out, but I think it rained the day before again, and then we went out again and took more samples of the lake.
Anything to add, Linda?
And it seems to always be higher when we sample out. So we also sample when it doesn't rain to compare, and it always seems to be higher. But I've read other studies that show, when there's precipitation, the levels are going to be higher. There's a really cool case study about Red Lodge and the Giardia outbreak in the 80s. And it's because of the Mount St. Helens eruption, actually.
Oh,
Excessive runoff
Okay.
from the snowmelt, and I wrote a cool lesson on it. If anybody's watching and wants that lesson, I can send it to you. But
there we
yeah, so
go.
it seems to definitely increase the levels.
Which would make sense, right? Because it's just going to bring more of the source into the lake from runoff when you have precipitation.
Mhm.
Let's see, are there any others? Okay, well,
One just popped up from Jocelyn Miller. Oh, we got another one.
here we go. Okay, what type of vegetation is found between the golf course and the lake? Are there any plans to reduce runoff?
I guess type of vegetation. I know that after the tornado, they vegetation down there. I can't
Hunter,
recall.
could you say that again? I don't, it's what didn't come through so well.
Oh yes. So after the tornado, I don't know when it was again, probably 2016, around there, 2016. I know that the plan was to just drain the whole lake and get the garbage out, which they did, and then they did restoration to the vegetation out there. And I worked for the parks, and last year, and we had to take out the stuff that was helping grow the vegetation. So, I mean, that I don't know exactly what's out there. I mean, there's a ton of plants and vegetation out there. There is plans to try and reduce the runoff. I mean, they have to drain it and reline the lagoon, or not the lagoon, the effluent. But also, they're going to try chlorinating it, see if adding a little bit more chlorination, because they're under chlorinating, will try to resolve this. And we might not see again the effect right away due to probably many years of the effluent running off into the fields and it's staying on top of the surface. So we might not see an effect right away.
But there are plans to mitigate the issue by the golf course?
Yes.
Yeah, they're requiring them to line the holding pond. So the effluent runs into a holding pond there, and then it's irrigated out of that. And it's just a little pond, but they're going to line that so it's not infiltrating through the soil. Because, I mean, I suppose it could get into the groundwater, so that's concerning. And I think the vegetation is mostly just grass, and it's mowed. It's kind of just an empty grassy area. There's no livestock or anything, but so.
Okay. Well, it's positive that there are plans and it's working and it's moving in the right direction, can we say, in terms of mitigating the problem. Another question that's come in, how does this affect the fish in the lake? I could speak to that on one level, but Linda or Hunter, any comments there?
Yeah, I mean, so the Baker Lake, when we have a little dam and it goes into a creek and it runs all the way to sandstone or whatnot. And in sandstone, there's a high level of mercury in the fish. And I mean, there used to be fish in the creek going along, but now they have disappeared. And this could probably be due to the fact that this is running off into the creek, and this also could be affecting different types of wildlife because they can come and drink from this creek running to sandstone. So it'd be interesting to test the fish at Baker Lake. I think that's something we should also look into doing just because a lot of people come here to fish and they catch it and they eat it, and it should probably be a public safety health issue just to test it, to see, because I know there's signs posted at Sandstone that there is mercury in the fish and not to eat too much of it, you know.
Anything to add, Linda?
I, I guess I definitely think it should be studied. I haven't read a lot of research about how it affects the fish, so.
Okay. Yeah, one thing that I can add to that is when you have these toxic algae blooms, they consume the oxygen in the water. And so if it's left unchecked and resolved, the end state of that process is called eutrophication, where all of the oxygen in the water is consumed and it becomes an anaerobic environment, and that's when the vegetation and everything dies. And some of you listening in, or I don't know, Linda or Hunter, if you've seen this, but if you ever see, say, trees that are underwater a little bit and they're all dead, it's one of the reasons that happens is this process eutrophication, where it's no longer an oxygen, there's no longer oxygenated water, and normal life forms will disappear. That's an extreme example. It's not what you have most likely currently. However, it's another reason to get after this and to not let this go unchecked. Any other questions from the audience?
I see two more that came in.
Here we go. Linda, do you or other Montana towns plan to reference this project for future research and water testing?
I definitely do. I think so one of the things that I want my students to learn is that science is for everyone, and it's a part of all of us, and it's a tool that we can use to figure things out. And I think this project has really shown that to my students and probably to the community too. Like science saves the day. We can solve problems and answer questions with science. So based on that, I hope that other towns or even teachers might try to tackle some of these questions, because some of it is like you have to have the confidence and the guts to pursue it, because it's a steep learning curve. It was a steep learning curve for both of us, right, Hunter? I mean, it was a lot to tackle, but we can do it. And I think it's a very cool opportunity to involve students. So I definitely think that there's a lot of potential for a project like this to be applicable to other towns in Montana.
Yeah, it's fantastic. I echo that. I think it's a wonderful model, and I would love to see it happen at other high schools, because, as they say, solving a real world water quality problem, it's real. It affects the public. It affects the community. At the same time, what a wonderful teaching opportunity and learning opportunity for the students to get in there, solving things that matter on a local level. And I just applaud you, for both of you, for your work and initiative and courage to take this on. I think it's really a great model to follow. Okay, another question. Has the upper lake been tested, and does the effluent reach the upper lake prior to lower Baker Lake?
Yeah, so the upper lake has been tested, and that would be on our little map, and that would be site E when we test it. And again, the golf course is 8 meters higher in altitude compared to the upper and lower lake, so it does reach into the upper and lower lake as runoff.
So it's both. It's flowing into both, and then they do release the upper lake if it's full, and so that would also flow into it. Yep, and we've been testing that. When there's water in the upper lake, we've been testing it.
And again, could you summarize, what do you find in the upper lake as compared to the lower lake in terms of coliform or whatever bacteria test that you conducted?
Mm-hmm.
You want to put that graph?
Yeah, I can go back to that graph here. Oh, all right. So the upper lake right here, again, you can see that it's not as high in coliforms compared to the other parts of the lake. But you do again have a high number of E. coli in there, and as you can see, that that the E. coli is about to surpass the geometric mean limit, and it's compared with the gazebo beach. Once it crosses that line again, they gotta shut down the lake. But the coliforms are not maxed out again, but they surpassed the statistical threshold as well.
Does the
Great.
next slide have the daily testing
Um,
or the next one?
this is the daily testing.
Does that show anything?
But yeah, on site E right here, you can see that on 9/6 or 9/7/2023, you can see that it's not as high of an E. coli compared to site D and A, but it does surpass the statistical threshold on this one.
Okay, great, thank you. Oh, one more question for you guys that I have, which is, what about other potential sources of contamination? Is that something you've looked at, thought about? Have any plans to take a look at other possible sources?
I mean, I know that before we got down to this one and really pinpointed it at the golf course, I mean, we looked at the possibility of other contamination such as livestock, but then there's no livestock nearby. And well, we just kind of went on, maybe there's a pipe broken, but you know, highly unlikely. Because I mean they weren't put in too long ago. But yeah, we've looked at other possible contaminations, but we found this one to be the main one.
The
And
main one
again,
to take a look at.
again, there might be other possible contaminations, but this is the main one right here.
Fantastic. Well, we have 2 minutes before we're at the end of our planned session. Any final comments from those listening in? Looks like we got all the questions. And I would just like to thank you both, Linda and Hunter, for putting this together for us. Again, applaud you for your pioneering research. It's just I get excited about this. I really hope to see more of this at Baker High School and other high schools in the state of Montana, and really any high school could get involved in something like this in their science programs. So great job, and thank you so much. Thanks everyone for listening in, and we're going to go ahead and wrap, and then have a great day, everyone. We'll see you next time.
Thank you.
Thank you.
Okay, bye-bye.